Hi everyone, welcome to Basic Science Series. I'm your host Lokendra Kumar, and today we will discuss types of hemolysis, which is alpha, beta and gamma hemolysis. If you have seen my previous video, I have discussed how to prepare blood agar. In this video, I will discuss how to inoculate the blood agar and how to detect the hemolysis on the blood agar and what are the distinct features of alpha, beta and gamma hemolysis. In my previous video, I have also discussed Staphylococcus aureus, where I have slightly, in a very brief manner, discussed beta hemolysis. But in this presentation, as I have already promised you guys that I will explain hemolysis in detail, I will explain alpha, beta and gamma hemolysis in detail. So I bring you this presentation today where I will explain these three important types of hemolysis. Before I start my presentation, I have a quick request to make. If you are new to the channel and you are watching for the first time, and if you like the video and if you like the content of the channel, then please do subscribe to the channel. It gives me motivation to provide cost-free education to everyone. And if you're a microbiologist, I have a lot of interesting videos that you can see, and definitely those videos are going to help you in your studies as well as in your exams. So with that note, I will just jump onto the presentation and I'll explain alpha, beta and gamma hemolysis in detail. All right, let's start the presentation. All right, let's start with this slide. If you have seen my videos, you know that I use my red pointers to explain things. So here I'm going to use this pointer to direct you to the various contents of my presentation. As you can see, I have designed these illustrations, and these illustrations, they are not real pictures. They are designed by me to explain the types of hemolysis. In this, you can see I have tried to design a blood agar plate where I have bacterial colonies that are present on the surface of the blood agar plate, and they are showing greenish zone of hemolysis. That particular greenish zone of hemolysis is mainly a characteristic feature of alpha hemolysis. That is why I have colored this alpha as green. So greenish to brownish color is the characteristic feature of alpha hemolysis. So if you have a bacteria on a blood agar plate, and surrounding that bacterial colony, you have greenish or brownish zone, that will indicate that you have alpha hemolysis. And second type is the beta hemolysis. Beta hemolysis is also known as complete hemolysis. Alpha hemolysis is known as partial hemolysis. I will explain in my later slide why they are termed as the partial as well as the complete hemolysis, right? Not the weak. Okay. In case of beta hemolysis, you will have complete hemolysis. That is why you will have clear zone of hemolysis. It will not be the greenish or brownish tinge in the hemolysis, right? That is why I'm trying to show you here. As you can see, the zones are clear. And in case of gamma hemolysis, which is a kind of self-contradictory, and also it's really weird because gamma hemolysis itself indicates non-hemolytic type of bacterial species, especially Anaerobic faecalis. So it definitely needs to be corrected. Maybe it needs to be updated. But right now, what is the case that gamma hemolysis indicates the hemolysis that is given by Anaerobic faecalis, and basically it's non-hemolytic, or there may be some kind of weak alpha hemolysis after 24 hours. But it's really self-contradictory that gamma hemolytic bacterial species, basically they are non-hemolytic. So I will now explain alpha, beta and gamma in detail. Basically now I have given you a basic idea of alpha, beta and gamma. Now we can discuss the concept in detail. All right, let's move on to the second slide. So right now I am on the second slide, and in this particular slide I will explain how to inoculate the blood agar and how to perform the analysis of the blood agar plate. Basically, what are the techniques that you use to inoculate the bacteria, and then what you can do to analyze the zones of hemolysis, right? And I have, as you can see, I have pointed out in these arrows, 4 arrows, there are 4 important points that you need to remember. Point number 1 is you need a fresh inoculum of the bacteria. So you need to grow bacteria overnight and use a fresh inoculum, as I have mentioned here. You can clearly see. And second point is inoculate the blood agar using your favorite sticking method. You can use a spread plate method, right? You can use a spreader and just spread the different dilution of the bacterial culture. It's recommended that you use different dilution of the bacterial culture, because then you will have clear-cut isolated colonies like this. So the zone of hemolysis, reading of the zone of the hemolysis will be easy, right? Or you can use this kind of streaking method, or whatever streaking method you like to use. And then finally, you'll have some colonies that are isolated and clear, and you can easily read the zone of hemolysis around those particular isolated colonies, right? So in this illustration, I have explained that you will have a bacterial culture which is overnight grown, and you need to grow the bacteria in 37 degrees centigrade or whatever the favorable conditions for those bacterial isolates or bacterial strains, depending upon the type of the bacteria, right? Anaerobic, aerobic, whatever bacteria you're using. After the growth of the bacteria on the media plate, you will use a light source, and you will basically hold your plate in front of the light source, so that using that light source you can detect the zone of hemolysis. Here I have tried to show you that the light is in the background, right? You can clearly see, and then I have a plate, and then basically I'm sitting in front of the plate and trying to see the zone of hemolysis. Basically, this is how you can read the zone of hemolysis. And if you have clear hemolysis, you can say it's beta hemolytic. If you have greenish or brownish color present in the zone of hemolysis, you can say it's alpha hemolytic bacterial genus or species. Now, so this illustration clearly explains the steps that you need to follow for the growth of the bacteria on the blood agar plate. And I have already explained how to prepare the blood agar and different facts about the blood agar. You should check that video. After streaking, you can read the hemolysis by using a light source. So basically, this is a protocol you can say that you can use to detect the zone of hemolysis on the blood agar plate. Now we will move on to the next slide, where I will explain beta hemolysis first, because it is also known as clear hemolysis. And first I want to explain the beta hemolysis, then I'll jump onto the alpha and gamma. All right, let's move on to the third slide. All right, here we are on our third slide, where I will start explaining beta hemolysis and start explaining the facts behind the beta hemolysis. As I have already explained, that beta hemolysis is known as complete or true lysis of the RBCs. You're using mammalian RBCs. Here, this illustration clearly shows that RBCs are getting lysed by the toxins produced by the bacterial cells, right? So as you all know, that I try to make these illustrations to make you aware of that particular concept in detail, because it's always very convenient to explain any concept using the illustrations. So basically what happens in this case, in case of the beta hemolysis, you will have a complete or true lysis of the RBCs. Second point is toxic byproducts that are released by bacterial samples that you have on the blood agar plate. In this case, the example is Streptococcus pyogenes. So you need to remember which Streptococcus species produces beta hemolysis. So Streptococcus pyogenes is the species that produce multiple kinds of toxins, And these toxins will lyse RBCs and produce the beta hemolytic zone. And I will explain what are the different toxins that are produced by Streptococcus species, specifically Streptococcus pyogenes. So as you can see, I have designed this illustration where I have bacterial cells, these round violet color gram-positive cocci, resembles the Streptococcus pyogene cells, and they are producing these triangles. These triangles are the multiple toxins that are produced by Streptococcus pyogenes. And I have told you that I will explain in my next slide what are these toxins. And then as you can see, when these toxins, they interact with the RBCs, they attack on the RBC membrane, and that causes the destruction or lysis of the RBCs. That is why it is also known as true lysis. In case of alpha hemolysis, you will not have the true lysis, and the lysis will be incomplete. That is why it's also known as partial hemolysis. Okay, so this was the basic concept of the beta hemolysis. Now let's jump on to the next slide, where I will explain what are the different toxins that are produced by Streptococcus pyogenes that leads to the destruction of RBCs and demonstration of the beta hemolysis. All right, we are in the next slide, where I will explain the different types of toxins that are produced by Streptococcus pyogenes. So you can see here, specifically I have mentioned Streptolysin O and Streptolysin S. These are the two different types of toxins that are produced under two different kind of environmental condition. One under the low oxygen level, and second one under the normal oxygen level. So when someone is trying to detect Streptolysin S, that particular analyst will grow the particular bacteria, Streptococcus pyogenes in this case, under the normal oxygen conditions. If the analyst or the researcher is trying to analyze trying to detect the Streptolysin O, the bacteria should be grown under the low oxygen level. I will say not specifically fully anaerobic environment, but maybe an incomplete environment, anaerobic environmental condition. So how you can develop or how you can provide anaerobic environmental condition is by using the stabbing method. So as you can see, I have designed this illustration here. What I'm trying to show you here is, this is the blood agar plate. So if you are streaking the bacteria using the inoculation loop on the surface of the blood agar, the bacteria will be grown under normal oxygen condition, unless you're not using any kind of anaerobic jar or any other anaerobic incubator or other technologies, right? For there are various kinds of technologies that you can use for anaerobic culturing. If you are not doing that, if you are using normal incubator and trying to streak the bacteria on the surface of the blood agar, the bacteria will grow under normal oxygen condition. But if you stab the blood agar, because you will have a thick layer of blood agar in the plate, right? If you stab the blood agar using inoculation loop, the loop which is full with the bacterial inoculum. So if you stab that, the loop will get inserted into the blood agar, and inside the blood agar, bacteria will get transferred. And that particular area where as you can see, the head of the loop is inserted, this area will be considered as partial anaerobic environment or low oxygen environmental condition. So if you want to detect Streptolysin O, you can try to isolate the bacteria present in this area and try to detect whether you have Streptolysin O present in the blood agar, or you can try to see the zone of hemolysis in this area. And if you have hemolysis, most likely the hemolysis will be due to the Streptolysin O. But if you have bacteria that is streaked on the surface of the blood agar, you will have the hemolysis because of the Streptolysin S. So in this presentation slide, what I'm trying to tell you is there are two types of toxins, Streptolysin O and Streptolysin S, and they both have different environmental conditions. And how you can detect the presence of Streptolysin O as well as Streptolysin S in your experiment is basically by using stabbing method or just culturing the bacteria on the surface of the blood agar plate. Then you can use the technique that I've already showed you under the light condition. You can try to see whether you have any zone of hemolysis. If you have zone of hemolysis, that will indicate that there is some kind of destruction of the RBCs is going on, right? So I hope that Streptolysin O and Streptolysin S is clear to you and how you can detect the hemolysis by these two different toxins. All right, let's move on to the next presentation slide. All right, in this particular presentation slide, I will explain alpha hemolysis. I have already explained what is the difference between alpha and beta hemolysis. Alpha hemolysis is slightly greenish. The zone of hemolysis is slightly greenish to brownish, and the beta hemolysis is clear. And why is that? The point number 1 explains that particular fact. As you can see, red blood cell hemoglobin gets converted into methemoglobin, and that methemoglobin is basically greenish to brownish in color, and it is unable to bind to the oxygen. And because of the color of this compound, the zone appears as greenish to brownish in color. Also, you can see here the iron is in F3 positive state, not F2 positive state. Because of this reason, the hemoglobin is unable to bind to the oxygen. Second point is green to brown discoloration. As I've already told you that the color of the media, which is normally red because of the presence of the RBCs, will change to greenish to brownish zone because of that incomplete hemolysis. Cell membrane is intact. In this case, the important point is that the membrane of the RBCs will remain intact, and because of the conversion of hemoglobin to methemoglobin, the RBCs, they will appear slightly greenish to brownish in color. Another point, it cannot be confused with the weak beta hemolysis, right? I'm not using weak hemolysis as the alpha hemolysis. I'm saying partial or incomplete hemolysis is the alpha hemolysis. So it is really important because there are some bacteria that will give you weak beta hemolysis. The examples are Streptococcus agalactae. Second one is Listeria monocytogenes. These particular organisms, they will give weak beta hemolysis, and Streptococcus mutans will give alpha hemolysis. So you can use Streptococcus mutans as a positive control in your experiment if you're trying to detect alpha hemolysis. If you have number of samples and you want to have a positive control, Streptococcus mutant is the bacteria that you can use, and you can compare your result. And these are the examples of weak beta hemolysis. So never get confused with weak hemolysis and say that the weak hemolysis is the alpha hemolysis, right? So weak beta hemolysis is not alpha hemolysis. Basically, incomplete or partial hemolysis is the alpha hemolysis. All right. So I hope you have now a basic idea about the alpha hemolysis. Now let's jump on to the gamma hemolysis. All right. So we are on the last slide of our presentation where I will try to explain. Gamma hemolysis basically means no hemolysis. Non-hemolytic bacteria, especially Anaerobic fecalis, that was initially the part of the Streptococcus and was known as gamma. So basically, within 24 hours, you will have no hemolytic activity. But maybe after 24 hours, you will have weak alpha hemolysis. So I don't know why it has been characterized as a gamma hemolysis, because there is basically no hemolysis up until 24 hours, and maybe there is some weak alpha hemolysis after that. So this definitely needs some kind of, maybe some update is required to rename this kind of hemolysis. But anyways, right now, gamma hemolysis means that there is no zone of hemolysis up to 24 hours, and especially this particular gamma hemolysis may show some reaction or a weak alpha hemolysis after 24 hours. Example in this case is Anaerobic fecalis. So this was a brief, brief introduction of the types of hemolysis, where we have discussed alpha, beta, and gamma. And I just want to summarize the same points that I have discussed. As you can see, I have that is why I have used these coloration. As you can see, alpha is in green color, indicating that greenish to brownish coloration is the characteristic feature of alpha hemolysis. In this case, it's clear. So beta hemolysis indicates that there will be complete or true hemolysis, and the zone will be clear. That is why I'm using white color for beta. Third, I'm using red color for gamma hemolysis, because there is no zone of hemolysis, and basically the media will appear as red, the same color of the blood agar, right? So I have also tried to explain to you what are the different examples of alpha, beta, and gamma hemolysis, how you can detect, what are the techniques that you can use. And I hope that this detailed video is going to help you to understand the types of hemolysis, as well as to know what are the examples and how you can basically detect the hemolysis in the lab. So with that note, I will just conclude this presentation, and I hope if you enjoyed the presentation, if you like the presentation, then please like, share the video with your friends. And if you like the content of the channel, then please do subscribe to the channel, so that I can make more interesting and more high quality videos, and I can put my 100% effort in my videos. So with that note, I'll just conclude this presentation, and I'll see you in my next video tutorial, where I will bring you interesting topic, different kinds of organisms that I'm going to cover. My objective is to cover most of the organisms, and maybe I will be able to make a complete bacteriology series that will benefit many students in future. So with that note, I'll just conclude my presentation, and thank you for watching this particular video. Please stay tuned. Thank you and take care.